Hi all. So in this video we're going to see about enzyme linked hormone receptors. So these are receptors to which an enzyme is linked for its action. So we'll see more about this. So enzyme linked hormone receptors are single transmembrane proteins. So see, when compared to your G protein coupled receptor which passes the membrane 7 times, here this is a single transmembrane protein and it has got a hormone binding extracellular part and an enzyme binding intracellular part. Okay. So based on the type of enzyme that is related to this receptor, you can classify them as receptors with intrinsic tyrosine kinase activity and receptors without intrinsic tyrosine kinase activity. So what is this tyrosine kinase? See, tyrosine kinase is a type of protein kinases. That means it can phosphorylate different proteins. So there are receptors which has got intrinsic tyrosine kinase activity and there are receptors without intrinsic tyrosine kinase activity, which means they'll have to depend on other cytoplasmic tyrosine kinases. Okay, so that is how we classify enzyme linked hormone receptors. So now we'll see each one by one. So suppose this is the cell membrane and this is the enzyme linked receptor. A good example for receptor with intrinsic tyrosine kinase activities are insulin receptor. So see, when the receptor is inactive, the the conformation of the receptor would be like this. That means they will exist as monomers. But once the hormone binds to the receptor, then these two subunits, the monomers, will come together so as to form a dimer. And this will cause a conformational change in the hormone receptor. So once the hormone binds to the receptor, it will cause a conformational change to the receptor, and this conformational change will activate the tyrosine kinase activity that is present within the intracellular domain. Okay, so it already has tyrosine kinase activity, so that will be activated. This in turn will cause autophosphorylation of the tyrosine residues. So that means the receptor would activate itself because it has got intrinsic tyrosine kinase activity. So there will be phosphorylation of the tyrosine residues. So now this part will act like an enzyme and it will phosphorylate other intracellular protein substances. Okay, so there will be phosphorylation of intracellular protein substrates by this intracellular part of the receptor which has got intrinsic tyrosine kinase activity. This in turn will cause enzyme activation as well as gene transcription, which will produce physiological effects. So this is how receptors with intrinsic tyrosine kinase activity work. So good example for such hormones that work through this pathway are insulin and epidermal growth factor. I will just summarize the effect once again. So the hormone binds to the receptor, which will cause a conformational change in the receptor. The intracellular portion of the receptor is exposed for autophosphorylation. Thus, the receptor will phosphorylate the tyrosine residues on itself. The receptor acts like a tyrosine kinase and phosphorylate the intracellular protein substrates. This in turn will initiate a cascade of activity leading to enzyme activation and gene transcription, and thus there will be physiological effects. So this is how receptors with intrinsic tyrosine kinase activity work. So what about receptors without intrinsic tyrosine kinase activity? So we'll just see an example for that. A good example for receptors without intrinsic tyrosine kinase activity is growth hormone receptor. So initially the growth hormone receptor would be in inactive state, but once the growth hormone binds to the receptor, there will be conformational change which in turn will activate, or it will open up the sites for docking of other tyrosine kinase like JAK, that is Janus kinase. This is also a protein kinase which is present in the cytoplasm. So when the growth hormone binds to the receptor, there will be a conformational change in the intracellular part which will open up the sites for docking of these Janus kinases. So once the Janus kinases bind to the receptor, it will be activated. That means they will be phosphorylated. This in turn will cause activation of other intracellular proteins like STAT proteins. What does STAT proteins stand for? They are signal transduction and activation of transcriptor proteins. Signal transducer and activator of transcription proteins. That is the full form of STAT. So these STAT proteins will now bind on to the receptor and they will be phosphorylated. So now see, once they are phosphorylated, they have become a dimer. And this dimer will be translocated and they will in turn will phosphorylate other substrate and also cause modulation of gene transcription, and thereby they will cause physiological effects. So this is how receptors without intrinsic tyrosine kinase activity work. So see, once the growth hormone binds, there will be a conformational change. The docking sites for JNS kinases are now open. So JNS kinase, which is a protein kinase, will be activated. This in turn will activate the STAT proteins, which when it gets phosphorylated, it will be a dimer, and this dimer will in turn phosphorylate other substrates and modulate gene expression and thereby cause physiological effects. Okay. So a good example for receptors without intrinsic tyrosine kinase activity are growth hormone, prolactin and cytokines. So now we'll just see this in a um, flowchart manner. Hormone binds to the receptor, so there is changes in the intracytoplasmic tail of the receptor, which exposes sites for docking of cytoplasmic tyrosine kinases like Janus kinase, and thus the docked tyrosine kinases are activated. They will phosphorylate the other cytoplasmic substrates like the STAT proteins, which in turn will cause phosphorylation of substrates and modulation of gene expression, thereby causing physiological effects. So thus we have seen the enzyme hormone receptors. We have seen how the receptors with intrinsic tyrosine kinase activity work with examples, and also receptors without intrinsic tyrosine kinase activity with examples. So I hope this concept is clear now. Thank you.